Ready for another exciting speaker, which is Vlad Vitoc, who is the founder of Maya Biotechnology, a company that develops novel agents for the treatment of a wide range of cancer types. Welcome.
Thank you. Okay, so thank you for coming to this talk. Maya Biotechnology is indeed an extraordinary company. We are the makers of the first telomere targeting agents. Thio is the lead agent. It's in the clinic now, and we are advancing a pipeline of next-generation telomere targeting agents. The efficacy of Thio has proven to be nothing short of extraordinary. We have seen curative effects when used in sequential combination with immune therapy, checkpoint inhibitors, in many tumor types. This efficacy was observed at dose levels 40 times under the maximum tolerated dose. Such a combination of efficacy and safety is unprecedented in cancer treatment. The FDA have reviewed this data and awarded Thio 2 orphan drug designations, one for HCC, hepatocellular carcinoma, the leading histology in liver cancers, 90%, and the other one in small cell lung cancer, the most deadly form of lung cancer. Regeneron are our partner. They provide their drug Libtayo, an excellent immune checkpoint inhibitor, under a clinical supply agreement for the phase 2 go-to-market accelerated approval Thio 101 trial that is going on right now in non-small cell lung cancer. This trial started enrolling a year ago in Australia and is expanding now into Europe, and later on comes to the United States as well. The upcoming milestones from this trial are near, and to include a few here: dose selection, overall response rates, the disease control rates, duration of response, and progression-free survival. A second phase 2 trial, also go to market, is called Thio 102. It's a basket umbrella design that is planned to start later this year, and this addresses several different tumor types. And the third, it's a phase 2/3 confirmatory and expansion trial, also set to start later. We have multiple telomere targeting compounds in development. 3 are already announced, and they started development earlier this year. Here's a look at the pipeline. Up top, you have the Thio 101 trial in non-small cell lung cancer, where we test Thio in sequential combination with Libtayo, the one that started enrolling in Australia a year ago, and is on track for filing for accelerated approval in the US by 2025. We are potentially just less than 2 years to commercial. The second trial, Thio 102, starts with colorectal, HCC, and solid tumors of all types as a signal generation arm. As soon as we see efficacy forming in a certain tumor type, we make it into its own arm. And the third is Thio 103, starting with non-small cell lung cancer and small cell lung cancer, both in first line of therapy, where most patients are. The second generation of telomere targeting agents, the first two, Agent 20 and Agent 12, has started the IND enabling studies about 6 months ago, are well underway, and on track to go to clinic next year. A third one was announced a month ago, will start development as well in the next couple of months. So Thio is short for 6-Thio-DG, or 6-Thio-2' Deoxyguanosine, the first telomere targeting agent. It's a small molecule, penetrates the blood-brain barrier, capable of treating brain cancers, eligible for NCE, New Chemical Entity, marketing exclusivity, the strongest you can get in the US and Europe and Japan, has a dual mechanism of action, telomere targeting, but also immunogenic effect. And we have seen complete response with no recurrence in multiple tumor types. The next generation of telomere targeting agents work through a similar mechanism of action, telomere targeting and immunogenic effect. And structurally, some are an evolution of Thio, others are new structures. We've tested them all in vitro. We created 84 molecules. We tested them all in vitro and then in vivo, and 7 of them proved to be an order of magnitude superior in certain tumor types. So the objective here is to advance to pre-IND development, 1 agent every 12 months. So cancer, unfortunately, is here with us for a longer time. It is the most dominant of the age-related disease. It affects the elderly, and the population aged over 80 is expected to triple in the near future. At age 90, the probability to be diagnosed with a type of cancer is 40%, and to die of it, 20%. By age 100, these numbers double. So yes, we want to live longer, but first, we have to not die of cancer. So thio is the only direct telomere targeting agent in development, and it works through a dual mechanism of action. First, telomere targeting. Second, immunogenic effect. So telomere targeting. Here to the left of the slide, in blue, you have the chromosome that contains our DNA, our genetic information, and at the end of the chromosome arms, in orange, are the telomeres. The telomeres are also DNA structures, and they have a very specific function to protect the integrity of the chromosome, specifically in the cell division cycle. And they are built by the enzyme telomerase that is present in all normal cells in the first year of life. At approximately age 1, telomerase goes away. And at that point, the telomeres have reached their maximum length. And from then on, with each cell division cycle, they lose a little, until in the old age, they become critically short, can no longer protect the chromosome, mutations begin to appear with the diseases of the old age, including cancer. But in cancer cells, something extraordinary happens. The enzyme telomerase is reactivated. And so the cancer cells regain their ability to lengthen their telomeres. And so they reach a state of replicative immortality. Just a scientific way of saying they continue to divide, the tumor grows in the aging body. And this is where thio comes in. Thio is picked up by telomerase, placed in the structure of the telomere, creates a faulty structure. The telomere collapses, and the cancer cell dies. This process is very fast and efficient. It happens in 24 to 72 hours. And thio directly kills 70 to 90% of the cancer cells. Then follows its immunogenic effect. Of the telomeric fragments, it forms micronuclei and present these fragments to the immune cells, triggering immune responses that are so strong that if you follow thio with an immune therapy, a checkpoint inhibitor, you get complete response, no recurrence, and anti-tumor immune memory. And we have seen this with thio in combination with Libthio of Regeneron, with Keytruda of Merck, with Tecentriq of Genentech, and the first go-to-market is with Libthio of Regeneron in non-small cell lung cancer. About our agreement with Regeneron, this is a clinical supply agreement under which Regeneron provide Libthio, cemiplimab, free of charge for our Thio 101 trial in advanced non-small cell lung cancer. This is a very big deal for us. The trial is budgeted for 184 patients, 12 months of therapy at $15,000 per month. It's a $32 million participation. So separately, we raised along the way $40 million by selling shares, but this is non-dilutive. Regeneron do not get a position in the cap table. In exchange, what they do get is development exclusivity in non-small cell lung cancer for the duration of this trial, estimated at 2 years, which means we cannot do a similar trial with a direct anti-PD-1 competitor, such as Keytruda, in non-small cell lung cancer. Any other combination remains open, and any other tumor type remains open. It's a very good deal. So here's a look at our Thio-101 trial. We have, as of the making of the slides, we had 35 patients dosed. We have many more now. We dose about 7 to 80 patients per month at this point, and this is accelerating. So this trial is testing Thio in sequence with Libtayo, cemiplimab, in non-small cell lung cancer in second line of therapy or later. So non-small cell lung cancer is the #1 cancer type in the world by mortality and by cells. It's a very insidious disease. It evolves without any signs or symptoms. By the time it's diagnosed, in more than half of the patients, it's so advanced, it's spread to other organs, it cannot be cured with current treatments. Typically, the patients get a targeted therapy. If there is a target, 30% of the patients get it. But the others get Keytruda plus minus chemotherapy, and that's it. Keytruda plus minus chemotherapy works a little. 40% response rates, 30% stable disease as best response, 30% nothing. Patients progress after 6, 7, 8 months, and then they go to chemotherapy, also doesn't work almost at all, or come to our trial. In our trial, we are testing 3 doses of Thio, 60 milligrams per cycle, 180, and 360. 60 milligrams is the curative dose in mouse translated to humans. The maximum tolerated dose in humans as monotherapy is 2,500 milligrams per cycle. This is 40 times under, unprecedented in oncology. We are testing higher doses as well, 180, 360, just to make sure we don't leave on the table any possible additional efficacy. But even 360 is still 7 times under the maximum tolerated dose. So because of this, we started our Part A safety lead-in with the highest dose, 360. The way we give it, we break the dose in 3, 120 milligrams per day, Monday, Tuesday, Wednesday. It's an easy IV, 15 to 30 minutes, just like Libtayo or any other checkpoint inhibitor. Then 1 day of break for the immune activation to take place, that's Thursday. And then Libtayo on Friday, dosed as approved. We don't change anything about how Libtayo is given. We just add Thio prior to Libtayo to make it into a regimen that works a lot better. Cycles are every 3 weeks. So we concluded Part A, safety lead-in, no dose-limiting toxicities. And now we are randomizing in Part B, where the goal is to select the best of the 3 doses on the basis of ORR, overall response rates, or disease control rates, and then expand the best dose arm to a regulatory size arm for approval in the US market and others. So we've seen in Part A an excellent safety profile. We tested the highest dose, 360. And what we have seen is far better than the current standard of care, which is chemotherapy. Chemotherapy is terrible, has an incidence of grade 3, 4, 5 side effects, 70 to 80%. This requires, grade 3 requires hospitalization, grade 4 ICU, grade 5 is dead. 5% die because of the treatment. Nothing like this here. This is what we see. Grade 1. Grade 1. The grade 1 is very mild side effects. And what they are? Fatigue, decreased appetite, blood pressure fluctuation. This is basically how I wake up every morning. So there was a report of grade 2, a report of grade 3. This is that were very quickly resolved. So no dose-limiting toxicities. But more importantly, we are now seeing the preliminary efficacy, survival data. The very first 2 patients enrolled in Part A of the study in Australia continue to be alive. Now more than 12 months. These slides were made a month ago. More than 12 months from treatment initiation. This is huge because both of these patients have advanced stage 4 metastatic disease and heavily pretreated. They already went through 2 lines of therapy before coming to our trial and progressed through them, including one with a checkpoint inhibitor. Which is even more extraordinary is they continue to be progression-free following their last dose for so many months, now breaking into a year, without any new treatment. This is amazing. In real-world practice, patients in this situation survive with therapy 3 to 4 months, and without therapy, they die in weeks. This is fantastic, much better than the standard of care. The second metric that is surfacing as we speak is disease control rate, DCR. At the time of making these slides, we had a data cut of June 23rd. This is almost 2 months ago now. At that point in time, we had 11 patients with at least 1 scan for efficacy. Of the 11, 9 had disease control. That is 82%. Compared to the standard of care chemo, which is 25 to 35% disease control. This is more than double the efficacy of the standard of care so far. Here's the swimmer's plot that looks at the preliminary survival data. We see at the top the Australian patients we talked about, and just about every other patient is passing through the average survival for patients who failed 2 or 3 lines of therapy. We will be reporting a major efficacy update soon at ESMO. ESMO is the European Society of Medical Oncology, second largest in the world. It's going to take place in Madrid this year, October 20th, 24. So the rationale for this trial came from this test we did in 2019, in which we looked at thio in sequential combination with Libtayo of Regeneron. No, I'm sorry, with Tecentriq of Genentech. And we looked at, this was a mouse model in which was conducted in 2019 and published in Cancer Cell in 2020. Cancer Cell is a very reputable journal. Only the best quality data gets in. So here, graph to the right, you see tumor volume on the vertical and days after tumor inoculation on the horizontal. There were only 2 cycles of administration, days 5 and 10. No other therapy was administered. So here you see in gray is the control arm, grows very fast without therapy. It's an aggressive tumor. In orange, the atezolizumab monotherapy arm, Tecentriq, grows just as fast, confirming it's immunologically cold tumor. In green, thio as monotherapy has an important effect. But in blue, the sequential combination of thio and checkpoint inhibitor with an extraordinary effect. Look at the delta between the blue line and the gray line. Huge. 60% complete response. To put things in perspective, Keytruda, in first line of therapy, the market leader in the clinic, has 2% response. Now, we have seen the similar results with thio followed by Keytruda and thio followed by Libtayo. So we then followed the 60% complete responses for 70 days. There was no recurrence. 70 days in mouse is like 7 years in humans. At this point, so the curative threshold is 5. At this point, they are cured. But we go above and beyond cure. And we re-challenge with 5 times more lung cancer cells and no additional treatment. There is no cancer take. Immune memory is in place because of thio. And with this extraordinary premise, we're going to non-small cell lung cancer, to the second line of therapy, where the current standard of care is chemo. Chemo is very toxic, grade 3, 4, 5 side effects, 79%. Overall response rates are low, 23%. Complete response, 0. Progression-free survival, 4.5 months. Overall survival, 10 months. Just terrible. For accelerated approval, the targets are 35% overall response and 6 months duration of response. And for standard approval, just a 20% improvement over the survival we see here is an approvable drug. A very high probability of success trial. So we have a biomarker of efficacy. It's called TIFS, an acronym, stands for telomere dysfunction-induced foci. These are these little yellow dots you can see the arrows pointing at that indicate the destruction of the telomeres by thio. They always appear. They're very well correlated with efficacy. And they reach a maximum after 48 hours from administration with a range between 24 and 72. And their formation indicates the on-target mechanism of action of thio. Thio 102 is our second trial. To start later this year, it has a basket umbrella design, a modern design. The FDA liked this type of design. We start with 3 baskets: colorectal, HCC, and solid tumors, all comers. And in each, 3 umbrellas: thio, followed by Tecentriq, followed by Libtayo, and by Keytruda. And the objective is to determine the best combination in each of the tumor types, expand to a pivotal size, and go to market. This trial is starting with 9 shots on goal, plus non-small cell, 10 shots on goal for accelerated approval. Most companies at our stage barely have 1. We already have 10. And in colorectal, we have seen our best results to date. Always complete response, never recurrence. Even when re-challenged with 10 times more colorectal cancer cells with no additional treatment. There is no cancer take. Immune memory is in place because of thio. And in colorectal, the situation is even worse. In salvage setting, where we are going, is also chemo, the standard of care, but response rates are under 2%. Progression-free survival, 2 months, and overall survival is 7. Another high probability of success trial. We have seen similar results in small cell lung cancer, graph to the left, thio with pembro. In HCC, graph to the right, thio plus atezo. These are the data sets that the FDA reviewed when they granted thio the 2 orphan drug designations. And earlier this year at AACR, we have seen the best preliminary preclinical efficacy to date. Thio, for the first time in an HCC model, primary liver cancer, with 100% complete response as monotherapy. But when combined with Libtayo, we see a very durable response. You see the graph, the upper graph. In green, you have the control arm. In purple is Libtayo, has an efficacy, delays the tumor progression. Thio in red as monotherapy, complete response, but not all of them sustain in the very long term. But the combination, thio plus Libtayo, just flat. 90 days. That's like 9 years. When we re-challenged with 2 times more HCC cells and no additional therapy, there was no cancer take. Even 210 days out, that's 21 years. This is definitive. Thio 103 will also start later this year. This is in first line of therapy, starting as a phase 2 to become a phase 3. We begin with 2 trials, with 2 arms, non-small cell in first line with Libtayo, and also small cell as an expansion in combination with EP and Tecentriq, atezolizumab of Roche and Genentech as the standard of care. And then we expand to pivotal phase 3 for full approval. So from an intellectual property perspective, the goal is new chemical entity marketing exclusivity. The strongest you can get, most robust, gives 7 years to market on the market in the US, 10 years Europe, Japan, and other markets, beginning with the timing of approval. And this is supported by a very robust and growing patent portfolio. We have 17 patents, 5 are issued, others are pending. The most recent is TIO as an immunogenic treatment strategy in sequential combination with checkpoint inhibitors, good until 2041. So our management team is very experienced. I'm a physician by my first training and businessman by my second, and have worked all my career in oncology, in pharma and biotech, about 50-50 commercial and medical. And I had the privilege to work on some extraordinary oncology compounds, some of them paradigm shifts in their day, such as Nexavar at Bayer Oncology. Nexavar was the first tyrosine kinase inhibitor in its day, and indicated for kidney cancer and HCC, where we are going with TIO. Nexavar peaked at $1 billion a year. Then Tarceva at Astellas for non-small cell lung cancer, where we are also going with TIO, $3 billion a year. Xtandi for prostate cancer, now at $5 billion a year. But not one of them had such a curative premise going into the clinic as TIO does, nor such an extraordinary safety margin of 40x. Here's a look at the cap table. We can comment this later. The investment opportunity, I'm going to click through the slides a little bit faster. But here's a look at the comparable companies. The markets, as you know and track, are very depressed right now. Our market cap is low. When you look at some of the companies, comparable companies, that with their valuations after they show their duration of response data, where we will be next year, this is what we see, between 0.7 and $4 billion. A major multiple from where we sit today. Turning Point Therapeutics is a particularly interesting case because this is a company based in San Diego that were acquired by Bristol Myers Squibb last year for $4.1 billion. The lead agent of Turning Point, repotrectinib, is a TRK targeting agent. TRK mutations in lung cancer, 1 to 2% of patients. TIO works in all cells that are telomerase positive. In non-small cell lung cancer, that's 85%. We will be there. So we have a number of major inflection points ahead of us. The first one is the IND filing. The second one, which enables us to open studies in the US as well, which is filed. The second one is are the preliminary efficacy data from TIO 101, which will be at ESMO this year in Q4. Then we open TIO 102, TIO 103, and next year we have progression-free survival, duration of response, other major inflection points coming from TIO 101, with filing for US approval in 2025 and then accelerated approval in the US. In parallel, we developed the next generation of telomere targeting agents, with the lead being about 3 to 4 years after TIO going to commercial. And with this, I'd like to thank you for your attention and take any questions. Thank you.
Thank you for your presentation. I have two questions. The stem cells are telomerase active too. Why do they not affect stem cells? And the other questions seems miraculous. Shouldn't be all telomerase active cancer cells be treatable?
Yes, indeed, all the telomerase active positive cells are treatable. In fact, TIO will kill any cell that is telomerase positive, including the stem cells. Its activity is dependent on telomerase presence. The difference is, in normal cells that are telomerase positive, like the stem cells and a few others, they're not telomerase positive all the time. They are sporadically telomerase positive, maybe 2 or 3 days in a month. If TIO catches them, TIO will kill them too. We don't administer TIO every day, so it catches just a few. when we see this in the side effect profile that you have seen there, there's a little bit of neutropenia, a little bit of anemia. That's it. If we were to give Thio daily, it will be much more problematic, right? But that's not how it goes. It's 3 days in a cycle of 21 days.
Congratulations, look like great data. I'm a neurologist by training, and I'm wondering, are you planning to go into areas where cancer is even more devastating, like glioblastomas or sarcomas?
Yes, glioblastoma in particular. We have 3 investigator-sponsored trials under consideration right now, and we have excellent preclinical data on glioblastoma in combination with radiation therapy and also with checkpoint inhibitors. Sarcomas, we are in discussions with a team from Hong Kong as well for sarcoma. Sarcomas are only about 40% of them are telomerase positive, so there's got to be a selection, but that's on the low end. So when you think about what percentage of cancer cells are telomerase positive, it's in aggregate 85%, some are 100%, like small cell, for instance. Others are 90% colorectal, lung cancer, it's 85% non-small cell lung cancer. Sarcomas are on the lower end, neuroblastoma on the lower end, 40%. Still much higher than the other targets. Yeah, thank you. Yeah. I have a question in the back.
Thank you for this very impressive data. I'm a clinician myself, and I saw a lot of patients having immune-induced side effects of the checkpoint inhibitors. Now you say you didn't see any of that. Can you explain mechanistically why that is?
We are keeping an eye on this type of effects. This is obviously an important side effect to track, both by Regeneron and by ourselves as well. We think that at some point that something may surface. We have seen, for instance, liver function increase in a couple of patients. We think that could be because of the immunogenic effect. But we also think that that is an indicator of efficacy, because in these patients, they were the first patients to respond to treatment. They responded very quickly. We will keep an eye on that.
Thank you very much.
Thank you.
Thank you. Thank you very much.
Thank you as well.